Hello, everybody. Picking up right where we left off, we're going to talk about our heart's physiology now, as opposed to the anatomy that we went over in our last video. So let's review again the blood flow through the heart. This is so very, very important. I'll say again. So from the superior and inferior vena cava, right? Superior vena cava, inferior vena cava, the blood, the deoxygenated blood flows into the right atrium. The right atrium contracts. The blood is pushed through the tricuspid valve between the right atrium and the right ventricle. Then when the right ventricle contracts, the tricuspid valve is going to snap closed and prevent backflow into the atrium. And the blood will be forced instead up the pulmonary trunk to the pulmonary arteries and to the lungs. Once the blood is reoxygenated in the lungs, it's going to return via the pulmonary veins from either side into the left atrium. When the left atrium contracts, it will force blood down through the mitral valve into the left ventricle. Then the left ventricle contracts, the mitral valve snaps closed to prevent backflow into the left atrium, and the blood is instead forced up into the aortic arch and to the rest of the body, okay? So how exactly does the heart coordinate this cycle? Here's another depiction of the cycle, our pulmonary circuit and then our systemic circuit, right? Actually, let's go ahead and label that, because why not, right? Which is sending blood to the lungs, which means it's deoxygenated, right? We wouldn't be going to the lungs if we didn't need the oxygen. And then this is the systemic circuit, aka sending blood, oxygenated blood, to the body, okay? So when the atria contract, they need to do that at the same time and allow all of their blood to flow down into the ventricles. The ventricles need to not contract right away, otherwise you might not get the maximum amount of blood out of the atria, right? But you don't want it to wait too long because we need to get that blood to where it's going, right? So there has to be a pause in between the atrial contractions and the ventricular contractions. This is where our SA and AV nodes come into effect. Let's come back over here. This is, again, complete anatomy. We're looking at here the electrical system of the heart, okay? So here is that synatrial node, excuse me. So this is the SA node right here. And down here is the atrioventricular node, or the AV node right here. So if I actually have one showing the entire innervation of the heart, we can see how that, well, actually we can't very clearly see how the SA node and the AV node are innervated, but we do know that it has a lot to do with the vagus nerve, right? So remember that cranial nerve from your brain, which controls so many important functions of vital organs throughout your body, also has its role in innervating the heart, okay? So the vagus nerve is a really important one. I'm not needing you to know exactly which nerves go to the SA and the AV nodes. So don't worry about that too much, or even the tracts in between the two nodes, like this internodal tract. We've got 3 different internodal tracts here. Internodal as in between nodes, okay? So between the SA node and the AV node, you have several tracts. That signal, once it comes from the brain, gets to the SA node. The SA node is going to trigger the atria to contract. And then it's going to continue, it's going to propagate that action potential down to the AV node. And then the AV node is going to pause. It's going to pause for that half second while the atria completely contract and squeeze out all the blood before sending the signal down all the way to the apex of the heart and causing the ventricles to contract from the apex up. So the heart contracts from, the ventricles contract from the apex up, okay? So the SA node receives the signal from the brain, passes it to the AV node. Meanwhile, the atria are contracting. The AV node pauses for a second while the atria finish contracting, and then it sends a signal all the way down to the apex of the heart, and then the ventricles will contract from the bottom up, okay? All these fibers have a special name. They're called Purkinje fibers, and we're not even calling it that here, but let's see if we've got Purkinje fibers coming up here. Okay. Oh, we do right there. Okay, so your pacemaker cells, that SA node and the AV node, this little guy right here, and this little guy, oops, right here. They are in charge of this rhythm that is your heartbeat, right? The lub-dub that we are all so familiar with. Incidentally, the actual sound that you're hearing when you're listening to the heartbeat is the sound of valves snapping closed, which is kind of awesome. So the lub is the semilunar valves closing while the atria fill the ventricles. And the dub, the louder dub, is going to be the snapping closed of the tricuspid and the mitral valves while the ventricles contract and send blood up through the pulmonary trunk to the lungs or up the aorta and to the entire body, the entire rest of the body. So we're going to look a little bit at the electrical system. Let's talk a little bit about these action potentials as they travel through the heart and ways that we have of measuring them and what these sort of graphical representations are showing us about that rhythm of the heart. So the membrane potential of the heart is literally the current on the surface of the heart, flowing through the surface of the heart, flowing through, if you will, the myocardium, through the actual heart muscle, okay? We can measure that electrical signal. And it's actually a series of action potentials. So it's not just one, we're not just following one action potential through the heart. We've got a series of them happening here. But this is simplified, okay? So you have a pacemaker potential happening with the AV node, that pause, right? Then you have a rapid depolarization, which is the contraction, pause, contraction, pause. So the pacemaker potential is basically a slow depolarization while the atria are contracting. After the pause, you have to have repolarization. I'm sorry, after the contraction, you have to have repolarization, as we learned in part A, and then again, when we talked about the nervous system and action potentials traveling through neurons, you have to have a repolarization, kind of a reset, right? Because all of these action potentials are still ions flowing back and forth across a plasma membrane, right? The plasma membranes of your cardiac muscle cells, right? Those cardiac muscle tissue cells. So just to reiterate, I'm just kind of like repeating generally what we already learned about skeletal muscle contraction, the neuromuscular junction, and action potentials as they travel down neurons in the nervous system. You have to have depolarization, and that is the actual action potential traveling, right? Depolarization means that you're having ions flood from one side of the membrane into the other, and you're making something happen, right? So that's the action potential, depolarization, going from a positive side of the membrane and a negative side of the membrane to them being roughly equal. Then you have the thing happen, right? The contraction happens, the action potential gets sent. Then after that action potential has passed over that part of the membrane of whatever that particular cardiac cell, then that membrane needs to reset. So it needs to pump the ions back across the membrane where they started from to get to that resting membrane potential, the resting membrane potential, okay? All right, and this time over here, they don't show you what that actually is, but it is in milliseconds, right? So this is happening very, very quickly. All right, so here's a fun little GIF that I found for you, and my face is blocking it, which is unfortunate. So moving my face over. This, if you stare at it long enough, you might get a sense of not only the blood flow through the heart and the order of the structures that it passes through, but the relationship between this rhythm, this cycle, and the electrical impulses that are driving it, right? So you have your little SA node here, your AV node down here. The SA node gets a signal and contracts the atria. Meanwhile, it sends that signal down to the AV node, right? There's a little pause. You'll notice a little tiny pause between this SA node lighting up and the AV node lighting up. That's while the atria are completely empty. And then that AV node lights up and sends its signal all the way down into the apex of the heart. And along all of these Purkinje fibers, which was how I learned it, basically all the little branches of this nerve that's innervating the ventricles, to get to every part of the cardiac muscle of the ventricles all at the same time, right? Because you want your ventricles to contract at the same time. Otherwise you have arrhythmia, you've got a problem, right? So I would like very much for you guys to know these as, I mean, I'll take subendocardial conducting network, but it seems more difficult maybe than Purkinje fibers. Okay, so Purkinje fibers are the little branching of this nerve, the atrioventricular bundle that is innervating the ventricles, okay? I do want you to know the atrioventricular bundle, which is coming from the AV node, the AV bundle, which goes from the AV node to where it branches in the interventricular septum. And then you have the bundle branches, and one bundle branch goes to the left ventricle and the other bundle branch goes to the right ventricle, and they both branch off into Purkinje fibers. So don't worry about what they're called in each ventricle. We're going to call them the same thing, Purkinje fibers. Okay. Of course, we need to know about our SA node and our AV node. And everything else, don't worry about it. This is also Purkinje fibers. If the other thing is easier for you, then more power to you and I will accept it. But Purkinje fibers is what I learned, so. Okay, this internodal pathway, by the way, is going to be those 3 internodal tracts, right? It's not showing it on this GIF, but there are 3, on this image, but there are 3 internodal tracts. Amazing that the GIF is better than this textbook image. What is the deal? We do have 3 internodal tracts there. In case that was weird for you, it's there. Okay. All right. So we got all of our stuff here, bundle branches, Purkinje fibers. All right. So yeah, stare at this for a little while and get an idea of this rhythm. It might be helpful. So here is this image again that I said we would come back to. This time we have a little bit more understanding about the nodes. So again, here's the SA node here on the superior surface of the right atrium. You've got internodal tracts here leading to the AV node. But before we get to the AV node, let's talk about how the sinoatrial SA node sends its depolarizing action potential across the muscle cells, the cardiac muscle of the atria, which are separated from the ventricles by like connective tissue, right? That signal doesn't accidentally get down into the ventricles. It might bleed over a little bit, but you have what's called the fibrous skeleton of the heart, and it actually separates electrically it actually insulates the two sides, the atrial side on top versus the ventricular side on the bottom. So your atria get that signal from the SA node, they contract, blood flows into the ventricles. Then meanwhile, those internodal tracts carry the signal from the SA node to the AV node. The torch gets passed. The AV node pauses for a second, and then it sends its signal down the atrioventricular bundle, the AV bundle, through the interventricular septum, down the bundle branches, which are these two guys right here, to the Purkinje fibers, so that the entirety of the ventricles, that portion of the heart, will contract all at the same time from the apex up, right? Hence these guys right here. Okay, AV node, SA node, really important. The AV node delays the spread of the electrical signal by 100 milliseconds. That's amazing, right? That's crazy. So a millisecond is 1/1000th of a second. So 100 milliseconds, 100,000ths, so a tenth of a second. If my math is weird. If my math is correct, then it would be like 1/10 of a second, which is pretty awesome. That's pretty amazing, right? It's only pausing it by that much, but that's the amount of time needed to maximize the efficiency of the muscle, of the heart muscle. And problems with either of these nodes, of course, is going to lead to problems with your heart rhythm. So literally arrhythmia and your heart's ability to function efficiently or at all, right? Which is scary, and why artificial pacemakers are a thing. And it's awesome and amazing that they are a thing, that even works. It's pretty badass. Okay. Let's go back into nightmare territory for a second and talk about autonomic innervation of the heart. Okay, so we already mentioned the vagus nerve, which comes from the cranium, right? And innervates a lot of major organs, including the heart. So instead of getting, we know this, right? So we know this part, going down to the SA node and the AV node. Pretty cool, right? This is, since we're talking about a cranial nerve, it is the parasympathetic innervation of the heart. So the part that comes from the vagus nerve, see the purple, parasympathetic. I guess we'll have to do magenta and green. Okay, so the parasympathetic, right? Cranial nerves have parasympathetic fibers. You have a long preganglionic nerve and a relatively short postganglionic nerve going to the nodes of the heart. Meanwhile, because your parasympathetic and your sympathetic nervous systems they innervate the same things and they're working in tandem, although antagonistically, right? The sympathetic portion of the innervation of the heart goes to the same place, right? Because we innervate the same thing. They just take turns having control over it, right? So the sympathetic part, as we know, we've got a short preganglionic nerve that's involving the sympathetic trunk, right? So remember that sympathetic trunk right next to the spine. So we have the short preganglionic nerves and then long postganglionic nerves going to the heart, specifically your two nodes, your SA and your AV nodes, right? And they're just going to do two different things, right? So the sympathetic nervous system, fight or flight, is going to tell your SA node and your AV node to pick up the pace, right? Pump the heart faster. And the parasympathetic nervous system, which hopefully is the one that's active more of the time for you, is going to literally keep the SA node and the AV node down. So believe it or not, going fast, the sympathetic nervous system is kind of the default. You need the parasympathetic nervous system to keep the heart calm when you're not in fight or flight mode, which is pretty cool. So the sympathetic innervation starts in the cardioacceleratory center. Cardioacceleratory, which is spelled wrong here. I will fix that. Cardioacceleratory. Making myself a note here. We still have green going on, I hope. Okay. That is a sympathetic nervous system. We are accelerating the heart using this part of the autonomic nervous system, right? It's descending through the spinal cord. It is exiting the spinal cord to the sympathetic trunk ganglion, asympathetic trunk ganglion, and then the sympathetic, the postganglionic sympathetic fibers go to the nodes, go to the heart. Meanwhile, or sorry, not meanwhile, but later, when you are in rest and digest mode and your parasympathetic nervous system is in charge, the cardioinhibitory center of your brain is going to send a signal down the vagus nerve and go to the same places and just tell a different story to your heart, which is, you can calm down now, slow down, take it easy. We're in rest and digest mode right now, okay? Starts cardioinhibitory. We are inhibiting the speed of the heart. Exits the medulla of the brainstem through the SA and AV nodes. So they both innervate the same thing. That's really important. And really, it's just autonomic nervous system stuff that we should know, right? Okay. All right, good deal. Here's another graphic illustration of a single action potential. So again, membrane potential on the y-axis, milliseconds on the left. We've got, let me change my pen color here. So we've got the depolarization happening really, really fast. You've got a plateau as we kind of slowly start to repolarize. This purple line is the actual force of the contraction itself. So the contraction is actually at its maximum after this little plateau of the actual action potential. And it takes between the moment of depolarization to the end of the contraction, not the end of the electrical action potential, for it to reset, that repolarization, for that reset to happen. So this is the absolute refractory period wherein you cannot have another contraction between this moment of depolarization and this of the muscle being starting to relax. The muscle has to be relaxed enough to even contract again. This is just kind of to help your understanding. You do not need to memorize this graph. Electrocardiography. So an electrocardiogram, or an EKG, is when you put electrodes on the body of your patient, and that can be it can be on the arms and the legs. It can be all over the chest. Like, there's different ways that you can do this, and then you basically detect the electricity traveling through the body from various different directions, which is kind of amazing. So you can actually test, like, from here to over here. You can test from here to here, and you can see what that action potential looks like. You can graph it with an electrocardiogram in the form of an electrocardiogram. So the little chart that you get, the little graph that you get, that's an electrocardiogram. Okay. An electrocardiography machine would make you an electrocardiogram. Okay. So you're basically getting a picture of the electrical function of the heart. You can see by this characteristic pattern whether or not the AV and the SA node are operating correctly, right? You have a little bump to start with, which is the atria depolarizing. So you might think of that as the lub. And then you have Q, R, and S. And I don't think they actually stand for anything. We're just kind of like going along the alphabet. PQRST. I'm not sure why they named them that, but the P bump, the P wave here, is going to be your atria depolarizing, which means that's the lub of your lub-dub heartbeat. And then this is going to be this little pause right here. Okay. Right there. And then dub, this is ventricular depolarization. It's huge. R is its peak. We have a very strong electrical signal for ventricular depolarization. And then you have repolarization. We fall below membrane potential. And then we get back to resting potential, and this is the ventricles repolarizing. Okay, so your atria are depolarizing while this is all happening, and this is your ventricle repolarizing. So this is not associated with a sound. Okay, you don't hear the repolarization of the ventricles, but this can be kind of correlated with the sound lub, and this can be kind of correlated with the sound dub, lub dub. So here is your classic EKG. Pretty cool, right? So your electrocardiogram is going to basically have this pattern over and over again according to the rate of the heartbeat of your patient. The P wave, again, atrial depolarization. PR, from P to R, is going to be conduction from the SA node to the AV node. And the QRS is ventricular depolarization, so ventricles contract. And then the T wave is when the ventricles repolarize. So you do get a little bit of a wave there. Because this was so darn big, right? That depolarization is clearly visible, so the repolarization is also slightly visible. Then you have a little pause, and then you do the same thing over again. And by looking at this pattern for your patient and reading the height of these waves, the P, the R wave, and the intervals, the length of the intervals between P and R, the PR interval, between Q and T, the QT interval, you will learn things. You'll be able to translate that into the condition of your patient's heart. Pretty awesome. So here's another visual representation of kind of the same thing. The SA node sends its action potential depolarizing the atria, right? The atria are going to contract. You have a little pause while the AV node receives the signal from the SA node. Meanwhile, the atria are now starting to depolarize, and the signal goes to the down into the ventricles through the atrioventricular bundle, the bundle branches, and then the Purkinje fibers throughout the muscle of the ventricles. You get your R wave, which is the ventricles contracting, and then the ventricles are going to end their contraction and repolarize. So the red is repolarization, okay? Red is repolarization. So that repolarization happens in the same pattern as that depolarization happened, right? Down these fibers and up. Repolarization happens at the bottom and up. Repolarization, that's the pause in between signals. This pause right here. Pretty cool, right? So here are some examples of abnormal EKGs, ECGs. You've got a normal one here for comparison, a junctional rhythm, which tells you that the SA node is not working. You don't have any P node. You do not have a P node. You do not, a P wave. So instead of having this nice little bump right here, you do not have a P wave at all, and you go straight into an R wave. So the AV node is in charge of pacing the heart by itself, which is not ideal because you need your atria to contract to get maximum blood out of the atria into the ventricles. Without the SA node, your atria don't contract, and therefore you probably have leftover blood hanging out in the atria by the time the ventricles are starting to contract, which just affects the efficiency, the efficacy of your heart muscle. A second-degree heart block. If the AV node fails to conduct some of the SA node impulses that was sent to it, you're going to have more P waves than you have QRSs. And the Q and the R is going to be very, very small compared to a normal R wave, right? So you usually have, according to this, 2 P waves for each QRS. So 1 P wave, 2 P wave. Maybe this is the 1 P wave, 2 P wave, QRS, T, P, P, QRS, T. And a ventricular fibrillation, when the electrical activity is disorganized, so you basically have some sort of arrhythmia, the action potentials are occurring randomly. So the SA node and the AV node are just kind of shooting off at random. You've got a chaotic, grossly abnormal ECG, right? As seen here, that's a big, big problem, right? So this is seen in an acute heart attack or after an electrical shock. So this is what an ECG might look like after someone's been struck by lightning or has a massive heart attack. It's just gone kind of haywire, right? Pretty scary. Okay, an echocardiogram is different. So this is the difference between an ECG and an EKG. Sorry, I was using EKG incorrectly before. So an ECG is an electrocardiogram. An echocardiogram, which I also have spelled wrong here. Got a little dyslexic there, obviously. And an echocardiogram is it's basically an ultrasound of the heart. Okay, so this is different. You don't have your little waves going on in your little chart. This is instead a picture made by sending like harmless radiation, but not really radiation, magnetic imaging off of the soft organ, the soft tissues of the heart. And this way, depending on what like angle you get in the ultrasound, you can actually view the valves. So this is actually a valve opening and closing, and this is a valve opening and closing right here, which is really, really, really cool. I would say this is maybe the left atrium into the left ventricle, going up the aortic arch through the aortic valve. Right, so that's your mitral valve and your aortic valve. How cool is that? And you do have a little ECG, kind of. I don't think that's a true ECG, but something to tell you where you are in the contraction. So not only do you get to see the electrical functioning, actually you don't. So in an ECG, you get to see the electrical functioning, while in EKG, you are basically getting to see the heart's structure and its function. Right? Okay. Lastly, or almost lastly, we have diastole versus systole. You may have heard about your diastolic blood pressure versus your systolic blood pressure. So your diastolic blood pressure is generally, it's when diastole means the heart muscle is relaxing. Your diastolic blood pressure is specifically when your ventricles are relaxing, okay? Your systolic blood pressure is when your ventricles are contracting, okay? So you have your diastolic blood pressure is the small number on top for your blood pressure, and the systolic number is the one on the bottom. And the systolic blood pressure is usually a much bigger number because the pressure produced by your ventricles contracting is much larger. The pressure is higher than the pressure through your blood that's going through your, pumping through your veins when your atria are contracting, right? Because if your ventricles are relaxing, that means that your atria are contracting because they go back and forth, right? So if your ventricles are relaxing, your atria are contracting, that doesn't create a whole lot of blood pressure through your veins and into the cuff, right? The blood pressure cuff. So your diastolic blood pressure is usually a smaller number, and your systolic blood pressure is going to be a higher number because that's when your ventricles are contracting and you get a big push of blood through all of your blood vessels, and your blood pressure monitor picks it up, right? Very, very cool. So here's ventricular diastole. You have relaxation of the ventricles, which means that the atria are contracting and forcing blood down. We're looking down here, down at the heart. You're looking at the tricuspid valve. I'm sorry, the tricuspid valve and the bicuspid valve, because this is the back of the heart and this is the front of the heart, open as blood flows from the atria down into the ventricles, right? So the AV valves are open and your semilunar valves are closed. Whereas in ventricular systole, when the ventricles are contracting, you will see that your atrioventricular valves are shut. They have snapped shut. That's that dub. As the ventricles contract, dub, these two valves shut. Do tricuspid, 1, 2, 3 cusps, and your bicuspid or mitral valve here. Those are shut while the ventricles contract, and instead the aortic and the pulmonary valves open and the blood is forced through there, either up the aortic arch to the body or up the pulmonary trunk through the pulmonary arteries and to the lungs. Pulmonary veins, excuse me, to the lungs. Right? All right, here's another depiction of the cardiac cycle. You can find videos of it all over the place. All you need to do is search it up. But here's one graphic visual representation of atrial contraction, atrial systole, which is the atria contracting. Systole is contraction, but specifically atrial systole, atria contracting. Atrial diastole, but ventricular systole, as the ventricles contract. And then ventricular diastole as the atria prepare to go back into systole and contract, right? Okay, so your diastole here for both chambers is that's that pause in between lub-dub, right? You're in a refractory period and you are repolarizing both right here. Depolarization of the atria, so SA node sends its signal. The atria are repolarizing here. The ventricles are starting to depolarize. Depolarization here, forcing blood to the body and to the lungs. This is your systolic blood pressure indication right here. And then repolarization of the ventricles while blood gets dumped back into the atria. And we go around again. I like that one. I think that's pretty cool. Ooh, this is cool. So here's a little clinical application for you. Your heart sounds. If you were to get a stethoscope and try to listen to your own heart, you would not hear it very well. You might be actually listening to the your blood pressure pumping through your ears. So get a friend maybe and listen to, see if you can hear the different valves of the heart. You can hear each valve best at a different location on the thorax. So you can get, you can hear your little your aortic valve up here on the upper right. You can hear your pulmonary valve on the upper left. Lower left will be your tricuspid and lower right will be your mitral valve. So that's pretty cool, right? And this is very, very specific about where exactly you would place that stethoscope, which is really important in what your primary care doctor or your cardiologist knows, right? That's what you have to know. Okay, and finally, actually almost finally, developmental aspects. Your heart originates from endothelium, which is part of the inner tube of the blastosphere, the early embryo. Your heart starts pumping at day 22, so that's like 3 weeks, right? And then it sort of develops in this convoluted shape. However, it has these openings, aka the foramen ovale, in between the two atria, because you do not actually need to send blood to the lungs. The lungs are still developing and you aren't breathing air. So You instead just kind of like pass blood round and round and round to the rest of the body because you have reoxygenated blood coming in from the umbilicus here, right? All right, and some common congenital heart defects to know. A ventricular septal defect. So a septal defect is going to involve the septum or the wall in between either the atria or the ventricles. The interventricular septum, of course, is the wall in between the ventricles, but you do have an interatrial septum, which we don't talk about as much, but you can have an atrial septum. Your ventricular septal defect is going to be a hole in the interventricular septum, right? So if you've got a hole there, then you've got blood flowing back and forth between the two ventricles, which means that you're going to have some already oxygenated blood going to the lungs, and you're going to have some deoxygenated blood making its way to the rest of the body. You're probably going to be feeling tired and out of breath if you have this a lot, right? Because your body's not getting purely oxygenated blood every time your heart pumps. It's getting like partially reoxygenated blood. So you've got blood mixing between the two ventricles, and more blood is shunted from left to right because the left ventricle is stronger. So you actually end up with more oxygenated blood going to the lungs, which sucks because you need to have a low oxygen concentration of the blood entering the lungs in order for gas exchange to occur effectively. So yeah, you're gonna feel like you're breathing through a straw. You're gonna feel like you can't breathe, or you're just very tired. You're not getting enough oxygen, right? Coarctation of the aorta. So it's basically a narrowing of the aorta. Okay. So if you have a part of the aorta that is narrowed, then you're going to basically have the left ventricle is going to have to work much harder to push blood past that narrow spot, which just makes it tired and probably thickens the walls even more, which actually decreases the capacity of the left ventricle. So there's a balance here. You want to have a nice, strong ventricle, but you don't want to like be putting too much pressure on it to get too strong. Otherwise, that myocardium gets so thick that you actually start losing capacity inside of the ventricle itself, which is a bummer, right? So narrowed aorta, coarctation of the aorta. And the third is tetralogy of fallout. So you've got a tetralogy of effects, tetra as in four. So you've got the first effect, which is that the pulmonary trunk is narrowed. So you basically have like a coarctation of the pulmonary trunk here. You may have the pulmonary valve may be like stuck closed. Hypertrophied right ventricle. So the right ventricle now is having to work too hard to push blood up into the pulmonary trunk, into the lungs. Therefore, the wall of the right ventricle starts to get really, really thick, which of course it should not be, right? So you're going to be impacting the capacity of the right ventricle here. Third, the ventricular septal defect that apparently comes with this is going to be adding to the tetralogy of fallout by doing literally what the ventricular septal defect does by itself, which is allowing a mixture of blood between the left and right ventricles. And then finally, the aorta is now going to be receiving blood from both ventricles as a result of the ventricular septal defect, right? So that aorta is going to get both oxygenated and deoxygenated blood. So not only do you have a smallish right ventricle that's working too hard to get blood to the lungs, but that blood is partially oxygenated because of the septum defect. And you have deoxygenated blood getting into the left ventricle, which is going to be passed to the rest of the body, which means that you're going to feel tired because your body cells are not receiving pure oxygenated blood, which is a problem, right? Okay, you guys, so to wrap it up, I just want to reiterate, take a look at your to-do list here. Read the announcement on how to make a study strategy. Do it now. We're only halfway through the semester. There is still time. We have 3 more exams. So we're not even halfway through our exams. take the time to make a strategy via the instructions in my most recent announcement. Set aside some time each day and just go through the steps. Fake it until you make it, just do the thing. I promise that it will help. And the idea, the whole crux of the strategy is that you are studying in small chunks frequently, which means you need to start earlier. So literally today, set aside, have your little time set aside. The same time each day is ideal, but if you can't do that, then schedule it and set an alarm, set a reminder on your phone. If you use a paper calendar, write it down and check it off to, because it feels good to check it off. But take that time to, on one day, read the chapter. The next day, watch one of these lecture videos. The next day, watch the other lecture video. The next day, do your homework. The next day, do the other stuff here in the additional resources part of your to-do list, okay? So after you've done this stuff, go back and start doing your dynamic study modules. If you find them to be helpful, try it. You don't know until you've tried it. I've got some flashcards here. I have some fun introductory videos. You might want to watch these first if you think that would help you. These are nice and introductory, and they're friendly and fun. They might be a good place to start for you before watching the lecture videos. If you didn't, then obviously it's too late. You've already watched the lecture videos, but maybe next week. I've got some art labeling stuff here for you. This is found in the study area of My Lab and Mastering. Okay, so I'm not finding this anywhere weird that you can't get a hold of it. All of these things come from the study area in My Lab and Mastering, which you can get to anytime. And then I've got some extra stuff. We've got a little free quiz that I found. Here's the Wisconsin University little interactivity that they have that's for free. This is an additional Labster simulation if you're into that. Kind of cool. This video shows how they do different electrocardiograms. Kind of interesting. And then this would be a great thing to look at before lab, actually. This practice anatomy lab. This is basically a slideshow of all of the different structures that you might see in your sheep heart on Thursday. Okay, so if you do this in order, your to-do list, as long as you do it all, you'll be good. Just do it in chunks, and you can do it in whatever order you think is best. I would save the homework until after you've done some other things so that you don't bomb the homework. But other than that, start wherever it feels good. Put a timer on for an hour. Stop when the hour is up. Pick it up the next day at the same time if you can. That daily practice, starting early, is the only way that your brain is going to be able to absorb this information. Okay. I want to show you real quick once more, in case you haven't visited recently. My Lab and Mastering, this link over here on the left, open it up. The study area is right over here whenever it loads. Come on, baby, you can do it. I believe in you. Here's the study area, right under the e-text. Of course, gotta launch a separate window. That's fine. Okay, so look at all this cool stuff that you have. And these are going to be stuff that it's broken down by the type of study help that it is and the subject matter. So your practice anatomy is broken down by cat, pig. The cat includes your sheep. So your sheep heart stuff would be here. And anatomical models, which are your actual plastic models if you want to look at them. If that helps you for some reason, then more power to you. But again, it's just going to be a slideshow, but you can also quiz yourself and make yourself a practical. This is a great way to quiz yourself, okay? So this is all available, it's all there under this in the study area of My Lab and Mastering. which is right here, okay? Your discussion with study tips, I just started. If you want to add to that, feel free. I'll add stuff as I find it or hear about it as well. Here it is. And that's all I got for you for today. I will see you on Thursday. I'm really excited to see you guys again. I hope you've all been safe and that you had a nice relaxing break once again. And yeah, come ready. Watch the videos, read the chapter, come to lab ready. You want to try to identify as many of the external and internal structures as you possibly can. So make yourself a list, print yourself out the images from the lab description, which is here. Whatever helps you. Figure out what helps you. Figure out how your brain works. This is important, and it's a skill that you can carry with you into the future, not just this class. You're going to be glad you did, okay? And with that, I wish you all a good evening, and I will see you all on Thursday. Bye-bye.